Welcome to the MSU Fisheries and Wildlife Club, dedicated to natural resources exploration and education.
My name is Emily Johnston. I'm a second-year PhD student in the Fisheries and Wildlife Department, and I'm working with Jen Owen in the Avian Health and Disease Ecology Lab. I've always been interested in human health and animal health, and so that was an interesting interaction to me. So that brought me to the disease ecology here in the Fisheries and Wildlife Department at MSU. Particularly zoonotic diseases, which are diseases that are maintained in animal populations but can infect humans. With my research, I'm going to look at tolerance and resistance in animals. So to do so, I'm looking at birds. There are 3 birds in particular that I want to look at. Those are gray catbirds. They're our kind of resistant bird. And then American robins appear to be more tolerant. In between, we have the northern cardinal, who can amplify the pathogen. They can get a lot of it in their system, but they also can get sick from it. The first step to looking at resistance and tolerance is we are going to get blood samples from birds and just test the innate ability of the blood to kill different pathogens. And then we're going to do some captive studies. To get tolerance, you have to be able to determine the level of health of an individual once it's infected and whether it gets sick or not. You have to have the animals in captivity. So we're going to have those 3 species, robins, catbirds, and cardinals, and we'll be infecting them with Borrelia burgdorferi, the agent that causes Lyme disease, and see how infectious they are to ticks, so how many ticks they're going to infect and for how long they're infectious. And that'll tell us a little bit about their resistant abilities and their reservoir competence, how good they are at being a reservoir for that pathogen. They'll be infected with West Nile virus, and that'll really be able to tell us more about tolerance and what kind of health impact these different species experience from the same level of exposure to a pathogen. Zoonotic diseases in particular are really important for us to look at. The biggest component of emerging infectious diseases are zoonotic diseases. They make up 60% of emerging infectious diseases. And out of all the zoonotic diseases, the majority of them have wildlife as reservoirs as opposed to captive animals or domesticated animals. Nobody's really looked at tolerance. A lot of people have done work with resistance and how animals can kill a pathogen or get rid of it or not die from it. But this aspect of tolerance, those animals that can survive an infection and just not get sick, is really important because those are the ones that are passing it on to other organisms or are escaping detection. Like, we don't even know that they're sick or that they have this ability to transmit because they're not displaying any illness. Well, we're always looking for help. I mean, no research is done by one person, individually. So particularly when there's field work and captive studies involved, we will be needing assistance with that. And if anyone's interested, they can always just contact me, and I can tell them what's going on at that time and what we might need help with.
But do you wanna walk us through your favorite ice cream at the dairy store?
Phew, I thought you were going to ask me something like political or something. I was worried. My absolute favorite ice cream at the dairy store is the Sesquicentennial Swirl. It is absolutely delicious, and I like to get a scoop of it or 2 with hot caramel on top in a bowl, and it's really delicious. Second to that,